Assalamu alaikum warahmatullahi wabarakatuh. So today we are going to talk about linkage analysis. So what is linkage analysis and why we do and why we need to know such analysis or about linkage analysis. So for example, a couple gets married and now the woman is pregnant and there is some kind of what we call autosomal or X-linked or sex-linked disease running in the family. So now this couple wants to know what is the risk factor for their baby who is going to be born, what is the probability. So if we only say that the probability is 50%, it is not a satisfactory answer for the couple. They need to know exact clear response so that they can decide whether to keep the baby or abort the baby. For this purpose or reason, we have the concept of prenatal diagnosis. So in prenatal diagnosis, we can do amniocentesis and chorionic villus biopsy, with the help of which we can check for mutant allele. Before we could just use pedigrees, and with the help of familial pedigree we would decide and just say the percentage of chance, and usually it used to be 50-50, but nowadays this is not enough. The patient cannot make any decision based on the result of the pedigree. We need to make more precise and clear decision and give them clear answer. So for that reason we are going to do prenatal diagnosis, in which we are going to use amniocentesis and chorionic villus biopsy material to check for the mutant allele. If a family comes with us which has an inherited disease which runs in their family, so ideally if we know the gene which causes the specific disease in that family, or the specific gene present on which chromosome, and also if we know the where is that specific gene located on that specific chromosome, it will be very easy for us to diagnose such disease. So there's a disease called Marfan syndrome. So in Marfan syndrome, the gene for fibrillin is mutant, and there will be it will because fibrillin acts as a scaffolding protein for collagen. So in case of Marfan syndrome, we have a lot of information about Marfan syndrome. We know on which gene the mutation occurs, on which number of chromosome that gene is located, and what kind of mutation it is. So for such disease such as Marfan syndrome, we can do direct gene diagnosis. So what is direct gene diagnosis? So in direct gene diagnosis, if we know what is the gene which causes the disease and on which number of chromosome that gene is present and what kind of mutation it is, in the laboratory we can make probes. So probes are basically nucleotide sequence which will have complementary nucleotide sequence to a specific nucleotide sequence present on a gene. So what does this mean? So for example, if we know what gene and number of chromosomes, what kind of mutation on the mutant gene or mutant gene we have, we can make a nucleotide sequence or a probe which is complementary to that. So when we drop that probe into the DNA sequence, it will go and bind with that mutant allele, and so we can hit that gene exactly in genetic material, and if they bind, we can know that yes, this what we call this mutant gene is present, and that is the reason for production of disease, and the future generation will also have the disease. So this is direct gene diagnosis. So for example, if this was our mutant DNA sequence on the gene, then if we drop a probe for this specific DNA sequence, it will go and bind to this DNA, and as a result we will know, okay, really there is that mutant allele is present, and for this person, for example in the case of Marfan syndrome, we will say he will have Marfan in the future. So we can do direct gene diagnosis. But sometimes what happens when a new patient comes to us with autosomal dominant disease, we can easily find out the pedigree for such patient and answer the result. But the result will not be very specific and the patient won't be satisfied with our answer. So when we don't know what is the gene which causes the disease, what location that gene is present on the chromosome, and what kind of mutation has taken place within that gene, we cannot do here direct gene diagnosis. So when we don't know the gene, we don't know on which chromosome that gene is located and what kind of mutation took place. Here we are going to do linkage analysis. So now before we go into the detail of linkage analysis, we have to see and understand the concept of genetic markers. So what are genetic markers? Genetic marker is a gene or DNA sequence with a known location on a particular chromosome, and it is used to identify the species or the individual. So that is what is a genetic marker. So genetic markers are landmarks which are present in all human beings' chromosomes, and these landmarks are slightly different from each other. For example, here star, man, girl, and a leaf. These we are graphically representing as different genetic markers on a chromosome. Same markers will be present on another what we call chromosome, but with slight differentiation. For example, here we have the blue star, here we have the black star. What does this mean? So if blue star, if we can think hypothetically that whenever there is blue star, C triple bond G is going to come 10 times. Okay, this is our genetic marker here. But whenever there is black star, C triple G, okay, bond is going to be formed 40 times. So they are the same genetic marker but with slight variation. So as we can see on the paternal and the maternal chromosome, we have different genetic markers. So these genetic markers which are specific, but these markers can be slightly different. For example, they will have different number of repeats. Here we had 10, here we had 20. For example, here we'll also have same. Okay, that is the concept. So it is quite possible that, for example, if we here had what we call blue star, which is 10 repeats, and here if we have the black star, which is 20 repeats, we will call them as different alleles, but they're alleles of each other, but they're different. Okay, and genetic markers which are as different alleles. Then it can also be possible that if we have a blue man here also blue man. Okay, so they will be having the same number of repeats, 40 because they are both blue, they are man and they are both blue. So if they are equal, we can say that they are same alleles. So for different alleles, we can remember that the concept is that nucleotide sequence is only different, as we had here C triple time G 10 times, or that is the blue star, and C triple time G for 20 times, that is the black star. So this is one of the what we call concept of genetic markers. So these genetic markers are normally present in the human chromosome, and they vary from person to person. Now this variation or DNA variation are actually harmless clinically. They don't produce any kind of problems, but such variations are present in the population and may vary from person to person. And these variation have already been, or these variations or genetic markers have already been studied by the scientists. So scientists have already studied these genetic markers, and they have given us their exact location on different chromosome and given us all their variants, and these are finally called as genetic markers. So genetic markers, basically they will mark the chromosome, and they won't have they are molecular fingerprints. They are also called as molecular fingerprints. So the gene marker analysis has already been done by the scientists, and all chromosome present will have multiple genetic markers, which we already know because of thanks to the scientists. And we should know the concept that these markers should be polymorphic. That means they should be slightly different from one person to another. For example, as we remember, this person has blue star genetic marker and this person has a black genetic star marker, so this means they are polymorphic. So now we are going to look another what we call concept, that is about linkage, related to linkage. So for example, if this is our disease causing gene and this is our marker number 3 and marker number 4, so defective number is gene is the green gene with a smiley face, that is our defective gene. So during recombination, it may be possible that this fourth genetic marker may go to other chromosome, and also we can see that this defective gene is closer to the genetic marker, this genetic marker. Okay, for example, this is genetic marker number 3 and genetic marker number 4. So disease causing defective gene is closer to what we call genetic marker 3. So hypothetically, if defective gene goes with genetic marker number 3, and wherever in the generation, that is father, mother, son or daughter, whenever this marker, okay, this orange color marker appears, the disease appears, it means that the disease causing defective gene and the genetic marker number 3, they are linked together. For example, now if we study the genetic material of Moon and his parents, we came to know that whenever in their generation this genetic marker, okay, for example, this green genetic marker, as you can see, it's near the defective gene, right? Whenever this genetic marker appears, the disease is also present. And when this genetic marker is absent, for example, the pink monkey, the disease is also absent because they are not linked together. So this is the concept of linkage between the genetic marker and the defective gene. So basically in linkage, we will not look for the gene because we don't know what is the defective gene, right? We don't know, but we know the genetic marker. So we look for the genetic marker which is linked to that specific mutant allele. So now we know what is what we call genetic markers and what is linkage. So let's come back to our linkage analysis topic. So linkage analysis is done when we cannot do direct gene analysis. Why we can't do direct gene analysis? Because we don't know what is the gene which causes the disease. We don't know on what chromosome this gene is located, and we also don't know what kind of mutation took place in this gene. So what we do is we try to see that this disease causing defective gene, is it linked to any pre-known genetic marker. So if we can find that it is linked to some genetic marker, then we don't look for the defective gene, we will look directly for the genetic marker if it's present in the newborn. The genetic markers are already known and they have been discovered, and we know exactly where they are located on the each chromosome. So now if that couple comes to do the analysis and check for the disease, but if the couple comes alone, it will be difficult to do the analysis. But if the couple comes with some family members, including those family members who are affected by the disease and normal family members, it will be easy to do the diagnosis, because then we can find in the affected individuals that which genetic marker is linked to that disease, and we can also find in normal family members that which genetic markers are not linked to that disease. So the normal people and abnormal people, we can find the genetic markers, and then we can locate if that defective gene is present on the newborn. So for example, if for this family we consider that whenever there is green genetic marker, the defective gene will be present, and whenever there is a pink genetic marker, the defective gene won't be present. So if in the newborn the green genetic marker is present, he will have the disease, and if in the newborn the pink genetic marker is present, we can say that the disease might be absent. So now even though we don't know the disease causing gene, but we know which genetic marker is linked to that disease. So we can say whether the patient will have the disease or not. So basic concept of linkage analysis is that if someone comes with for genetic analysis, and and if we don't have the information about the mutant gene or the defective gene, or on which chromosome this gene is present, or what kind of mutation has taken place in this gene, we cannot do the direct gene analysis. We have to go for linkage analysis, and in linkage analysis we are going to check hundreds of genetic markers to look for linkage with disease. And if we locate the marker which is moving along with the disease, then we can say that that genetic markers is linked with the disease causing gene, and so we will look for that genetic marker in the future generation to diagnose that the future generation will have the disease. Locus is a specific position on a chromosome. So for example, the scientists have mapped these genetic markers. So so if we say at this position this genetic marker is present, so that means at this locus this genetic marker is present. So basically locus means specific position on a chromosome. So genetic marker is good when it is close to the disease causing gene, okay, but if the genetic marker is far away from the disease causing gene, it is not good, because that genetic marker might separate from the disease causing gene during meiosis and our diagnosis will be wrong. For example, here we have genetic markers like star, man, and this is a defective gene car and the flower. So the defective gene is closer to the car and the defective gene is far away from the flower. So if we determine that whenever there is a car in the family, the defective gene is also present and the disease is present, so our analysis can be more accurate. But if we consider that flower is closer, flower is the genetic marker which when appears we will have the disease, then we can be wrong, because during meiosis this genetic marker might go to another chromosome during recombination, because they are very far away from each other, that is genetic marker and the mutant allele or the defective gene. And this distance between the defective gene and the genetic marker is known as theta. So theta between the CAR genetic marker and the defective gene is small, so it is a good genetic marker, whereas the theta between the defective gene and the flower genetic marker is larger, so it is not a good genetic marker. The best way to know the theta between genetic marker and the defective gene is to look at the meiotic events, such as when crossing over takes place, that is recombination takes place. So how can we find out which marker is close? To find out which marker is close, we are going to do pedigree analysis. That is, we are going to do look for the markers within the DNA sequence. So if the disease causing gene and the genetic markers which we think is the suspected genetic marker are co-inheriting, okay, that means whenever CAR gene is present, the disease defective gene is also present, we will say that they are close or the theta is small, that is the interlocus distance between them is small. But if they are non-co-inheriting, this means that the distance between the defective gene and the genetic marker which we are considering is large and the theta is large. So if we have less theta, it is a good marker. So theta is the distance basically between the defective gene and the genetic marker which we are considering. So if the theta is large, it is a bad marker, but if the theta is less, it is a good marker. So how to know in laboratory if the theta is less or more? We will look at the familial tree, and if the marker which we are suspecting, the suspected genetic marker and the defective gene are co-inheriting, this means that most of the time the theta is small. And if the genetic marker which we are suspecting and the defective gene are segregating from each other, this means that theta is large or more. So for example, if we suspect a particular genetic marker and we think that this is a genetic marker linked with the disease, but sometimes when we see the genetic analysis, the genetic marker is present in the person but the disease is not present, so this means that theta was large and they are not close to each other. It is the defective gene and the suspective genetic marker. Now coming back, now we know what is theta, what is the advantage if the theta is small or big. Now the question is, how can we determine what is theta, okay, or is the theta small or large? For that, the answer is that the theta depends on recombination during meiosis. So to understand that, we are going to see a pedigree example, but before we see the pedigree example, we have to understand the concept of microsatellites. So microsatellites are genetic markers which are characterized with what we call, they have the same type of repeat, but the number of repeat varies from person to person. And also the microsatellite is present on the same locus on each chromosome. So what does this mean? So basically microsatellite, as we can see here, for example, this had they have a particular repeat, for example ATG ATG ATG. So here the number of ATG repeats is less in genetic marker number 1. In genetic marker number 2, the number of ATG repeat is even more than 1, and in genetic marker number 3 it is more, and then in genetic marker number 4 the ATG repeats are more. So they all are microsatellites, okay, because they have a particular type of repeats which is present on the particular location or locus on the chromosome, and it varies among the chromosome and from person to person. So here we can say that 1 2 3 4 genetic markers are microsatellites basically, and these are different alleles of the same microsatellite, so they have different variation. That is why they are called as different alleles of what we call microsatellite. And then those markers are very good markers, or those genetic markers are very good genetic markers which have multiple alleles, for example as we saw here in the case of microsatellites, because if a specific genetic marker has multiple variation, it means it can help us to look at chromosome of different people and helps us to tag that marker or to identify that particular genetic marker. So now that we have understood the concept of microsatellites, Coming back to our question, and that question is how to determine theta, and whether how to understand if theta is small and large. For that, as I told earlier, we have to understand, we can understand that through a pedigree example. So for example, if we look at this pedigree, here is the diseased individual and these are his chromosomes, and here is the healthy individual. So the female who is the diseased, in this she has here a diseased gene that is responsible for expression of disease. So this is an example of autosomal disease which we are going to see. So during meiosis, what happens that the chromosomes will be replicated, will replicate their genetic material. Therefore, here if we consider these purple ones as genetic markers, so genetic marker 1, 2, 3, 4, which are all alleles of each other, which we already saw as microsatellites. Okay, as a result, these genetic markers and the mutant allele will also be doubled. So let's see that here. So here we can see that this is the disease causing gene and here are the normal genes. So on this chromosome, if we see, this is this chromosome, and if we see this chromosome, this is this chromosome here. So the disease causing gene is the star and the normal allele was a ball. And then we had the genetic marker here which we saw in purple. Here I have drawn them in blue here. So genetic marker 1, and this is also genetic marker 1, but this is the allele 1 of genetic marker 1, and this is the allele number 2 of genetic marker number 2. Then we have another genetic marker number 2, which will this is labeled in purple color, and this is the allele 1 of genetic marker 2, and this is the allele 2 of genetic marker 2. Both of them are purple color. So what happens during meiosis, as I told, the chromosome will replicate the genetic material and genetic markers, and the mutant allele and the normal alleles will also be doubled. So here during meiosis there was replication of the chromosomal material. So here is the replicated chromosome. So this disease causing gene will also be replicated, whereas the healthy gene will also be replicated. Okay, or everywhere we can see them. Now here we can say that the disease causing gene is more linked to genetic marker number 1, which is in blue color, as compared to genetic marker number 2, which is in purple color. Now after the genetic markers and genetic material has been replicated, there will be crossing over of or recombination of genetic material. So here in this example we can see that from here the crossing over will take place, okay, or recombination of genetic material. So the result of this will be that the disease causing gene will be present in the original allele and in the copy and the normal allele. So the genetic marker 1 is present again close to it, but the genetic marker 2, there is some recombination has taken place, that is its second allele has appeared here and first allele has gone there. So they have shifted from here and from here, so they have gone like that. So this is the recombination which took place. Now to better understand the concept of theta is small and large, we will take the comparison of two different disease causing genes. So this in red star is disease 1 causing gene, and the yellow ball is the disease 2 causing gene. And then we have genetic marker 1 and its allele 1 and allele 2, which are in blue color, and genetic marker 2, which is in purple color, and its alleles are in that is its allele 1 and 2 are also in purple color. So here I have drawn a fresh diagram in which I am going to show you again once again. Here the disease causing gene, disease 1 causing gene is black star in black color, and marker 1 is again in genetic marker 1 is in blue color, and the disease 2 causing gene is a green star, okay, green color, and genetic marker number 2 is again in the pink or pink color or purple color. So after recombination, we had told the recombination took place and the second allele of genetic marker 2 came here and the first allele of genetic marker 2 went there. So this all happened after the recombination. So now let's see what will be the possible recombination or possible genetic material in this woman's ova, because we remember that the diseased person was a woman, so that's why we're talking about the ova. So let us see that. So we will see in the first ova, the star, the blue, the star will be there, that is disease 1 causing gene. There will be the genetic marker 1 will be present. Then there will be disease 2 causing gene, that is the green star, that is also present here. And then the genetic marker 2 allele 1 will also be present. So both the first alleles of genetic marker 1 and genetic marker 2 along with disease causing gene, disease 1 causing gene and disease causing 2 gene are present in the first ova. In the second and third ova, there is a slight difference. So as we know, the second ova will be made of this chromosome and including this genetic, and the third ova will what we call include this genetic material. So when we see there, we will see that the disease 1 causing gene is still present, and genetic marker 1 also is still present. close to the gene disease causing gene 1. And then in what we call the disease 2 causing gene is also present, but here the first allele of what we call genetic marker 2 is not present. Instead, the second allele of genetic marker 2 is present. And in the third ova, the what we call healthy allele is present of the mutant allele, and then the second allele of the genetic marker 2 genetic marker 1, sorry, is also present. And then we have the healthy gene of the mutant allele present, and unfortunately, which we thought that the genetic marker 2 second allele should have been present, but here the genetic marker 2 first allele is present, because we thought that genetic marker 2 second allele is linked to the normal gene, and the genetic marker 2 first allele is linked to the second disease causing gene. But here that was not the case, and we saw it is different, the second and third ova. In the fourth ova, everything is going to be normal. The what we call the disease 1 causing gene normal allele is present, and then the genetic marker 1 second allele is present, and then the disease 2 causing gene normal allele is present, and then the disease what we call genetic marker 2 second allele is present. So this is a normal. So here what we can conclude that in the first and the fourth ova, there was no recombination. Here and here, no recombination took place. So they are non-recombinant variants. Whereas in the second and the third, we had recombination which had taken place. And so we will call them the recombination. And not only this, in all the 4 examples, we can notice that the disease 1 causing gene, okay, it is always linked to the genetic marker 1. But the disease 2 causing gene is linked to different alleles of genetic marker 2. So this means that the black star, that is the disease causing gene 1, and the genetic marker 1, they have small distance because there were less recombination, whereas the distance between the disease 2 causing gene and the second genetic marker was large distance. That is why we could see their recombination. So this means that theta is found out basically by recombination, which can be seen in the pedigree analysis. And if there is more recombination, like how we saw here, that means theta is large, and if there is less recombination, theta is small. And theta is basically interlocus distance, that is the distance between the disease causing gene and the genetic marker which we are considering. And so we cannot see the distance between the genetic marker and the disease causing genes in a chromosome, but we can determine the frequency of recombination in product of meiosis. So how to determine the frequency of recombination in product of meiosis? To determine the frequency of recombination, we have to divide the number of recombination by total number of offspring. So here we had 4 offsprings and 2 recombinant variants. So that is going to be 2 by 4, that is equal to 1 by 2, that is equal to 0.5. That is the frequency of recombination for the case of green, that is the disease 2 causing gene, and the genetic marker number 2, that is the frequency of their recombination. That is 50% chances of segregation. And if you would like to calculate the frequency of recombination of disease 1 causing gene and the genetic marker 1, that is, we will see that how many number of recombinations divided by total number of offspring. So here the number of recombinations was 0 and the number of offsprings were 4. So 0 by 4 is 0. That means that the frequency recombination is very, very low. And we have to remember that frequency of recombination is equal to theta. So what is frequency of recombination? Frequency of recombination is theta. So if theta or frequency recombination is 0, that means the distance between the disease causing gene genetic marker is very small, and we can say that they are linked very closely to each other. And so if anytime we will see this genetic marker, we can say that the disease causing gene will be present in the offspring. So this was basically the concept of linkage analysis, in which we are going to use genetic markers to identify whether the disease causing gene will be present in the offspring or not. And that is determined by theta, and theta we can come to know by pedigree analysis. Now we are going to discuss about genetic markers and its properties. So genetic markers has 2 main properties, that is polymorphism and that genetic markers should be numerous. So what we mean by polymorphism of genetic markers is that, for example, if we consider the human face as a genetic marker for now, every one of us has nose, eyes, ears, but we all look slightly different. That means we all are polymorphic according to our face, if we consider our face as a genetic marker. We want our genetic markers to be polymorphic and not monomorphic, because it is easier to what we call associate the disease causing gene with the with such polymorphic genetic markers. For an example, if we consider a genetic marker which has only two alleles, we will not think it as a good genetic marker, because, for example, in world population, if only two faces are there, okay, then how can we know out of all the people in the world, okay, for all have no faces, only two people have face, then how can we know that who took our money? So that's the reason. So if we have different what we call genetic variations of the genetic marker, different variations, so then we can identify the disease easily. Or if every person in the world has slightly different faces, then it is easier to identify who took our money. So this was the concept of polymorphism. Now we will see the concept of genetic marker being numerous. So genetic markers property number 2 is that they should be numerous. This means that there should be many different types of markers, and each of these markers should have many alleles. So then that we can easily map the chromosome with these markers. So this is the main reason for what we call numerous number of genetic markers. Now we are going to discuss about the type of genetic markers. So we have discussed the property of genetic markers. Now we will see the type of genetic markers. So we have three main types of genetic markers: RFLP, SNP, and VNTR. And VNTR is of two types: microsatellites and mini satellites. So let us go in detail of all this. Before we discuss further about the genetic marker, types of genetic marker, we should know that our DNA is of two types: single copy DNA and repetitive sequence DNA. So single copy DNA means, example, insulin gene, we have only one insulin gene, or it has a single copy which can be seen here in DNA. And then we have repetitive sequence DNA. Most of our DNA is repetitive. We should remember that. This means that repetitive sequence DNA means some sequence of DNA which is being repeated again and again. So for example, here we can see, we can represent it as purple. So if this is a DNA sequence, in that this is our single copy DNA insulin gene, and this is our repetitive sequence DNA. One such example of such repeated sequence DNA, it is called as palindrome. Now we have to remember that this palindrome sequence, which is here in pink color, it can be present on different site on different chromosomes, that especially on male and female chromosome or mom and dad chromosomes. So here, if we consider this as a maternal chromosome and paternal chromosome, here we can see our palindrome sequences in pink, and here in pink also are the palindrome sequences, and both the chromosomes have the palindrome sequence at different places, but the main thing is that it is repeating, so that's why it comes under the repeative sequence DNA. Another thing to remember here is that in palindrome, restriction endonuclease can attack. So at these points, restriction endonucleases can attack. So for example, if we see here, our pink ones are the palindrome, so the restriction nucleus endonuclease can attack here and here and remove this fragment. And since it is endonucleus, it can attack in the middle, and exonucleus, as we remember, only attack at the edges. So here in the maternal and paternal chromosome, we consider palindrome. If we consider the palindrome as a genetic marker, we need to remember that these palindromes can be attacked by restriction endonucleases. And as we can see, the palindromes are located at different locations in the maternal chromosome and paternal chromosome. Both the maternal and paternal chromosome are homologous to each other. That is, both of them, for example, if you consider this is chromosome number 1, this is also chromosome number 1. They are homologous chromosomes, but the palindromes are located at different sites or locus on both the respective chromosomes. So now if we apply the what we call restrictive endonuclease to these chromosomes, they will form different length of fragments. For example, here there will be a different fragment, here there will be a different fragment, because the distance between these palindromes is big, and here the distance between these palindromes is small. And if we consider that the mother was what we call diseased, or she has the disease, she's carrying the disease and manifests the disease, so we'll see a mutation here, for example in her insulin gene. But in the paternal gene there was no what we call mutation. And not only that, the length of the paternal fragment is smaller and the length of the maternal fragment is longer. And this fragment which is formed is called as RFLP, that is restriction fragment length polymorphism, and it is a type of genetic marker. So we can use this concept as the mutant allele is present in mother. The mutant allele the present in mother, it has a longer length or a larger fragment. And in the paternal, there is a shorter length and a shorter fragment. So whenever in family the larger or the longer fragment goes, the disease will be manifested. And if the shorter fragment comes in the baby, the disease won't be manifested. So here, if we consider this to be the mutation, we can see the fragment is longer compared to the paternal gene where the fragment is shorter. So in what we call RFLP, the length is different in different people at different chromosomes, because different people have even if homologous chromosomes, the what we call the palindromes will be located at different positions. So length acts as the genetic marker. We don't know the gene, but we know that the longer fragment is associated with the disease in this example. So this was our first type of genetic marker. Now we are going to see the next type of genetic marker, which is called as SNP, here in green, which is represented here in green in this diagram. The full form of SNP is single nucleotide polymorphism. So this means that in different people at different loci, single nucleotide polymorphism are present. So here we can see in green SNPs, and here also. So as we can see, they are located at different position on the homologous maternal and paternal chromosome. The chromosome is marked by single nucleotide polymorphs at different intervals. This interval is different in different people. Example, if we take the chromosome number 1 of many people sitting in a room, the SNP will be repeating again and again, but it will be different from person to person. That means it will be located at different positions. And this is the second type of genetic marker. So our first genetic markers were the RFLP, that is restriction fragment length polymorphism. The second type of our genetic marker is the one in green, which is called as single nucleotide polymorphism. The third type of genetic marker which we are going to see is located over here, and it is called as VNTR, that is variable number of tandem repeats. Here also we can see these are two types of variable number of tandem repeats, or VNTR. So VNTR is basically specific sequence which repeats again and again tandemly. So tandem means one after the other. SNP and palindromes are also repetitive what we call DNA sequences, but they are not tandem, they are dispersed, that is they are located at different position. But variable number of tandem repeats are repeats which occur one after the other. So in this case we should not confuse between VNTR, SNP and palindromes. SNP and palindromes are repetitive DNA sequence, but they are dispersed, they are not located one after the other. Same goes for the single nucleotide polymorphism SNP. These variable number of tandem repeats can be considered as train which has many bogies or boxes. Variable number of tandem repeat is of two types. It has two types of satellites in it, that is mini satellites and micro satellites. So satellites are special area of DNA which vary from person to person. Usually they are harmless area but make the genetic code polymorphic. Okay, so we have two types of satellites, micro satellites and mini satellites. So if we consider variable number tandem repeat as train, so we have two types of train, mini satellite and micro satellite. So in mini satellite train, the each box is going to be around 15 to 70 nucleotide long, and the total length of this full train, okay, with all the boxes is going to be 1 to 3 kb length maximum. Okay, that is a mini satellite. Micro satellite is a small train with small box. So its each box is going to contain 6 to 15 nucleotides only, and the maximum length of this train is going to be 1 kb, not more than 1 kb. Mini satellites are mostly near the telomeres, whereas microsatellites are located throughout the chromosome. That is why microsatellites are better genetic markers as compared to mini satellites. So this was the third type of genetic marker. So here we can see the third type of genetic marker, that is the microsatellites. And here is the mini satellite that is not more than 1 kb in length, and this is 1 to 3 kb in length with bigger boxes. So bigger train with bigger boxes, smaller train with smaller boxes. These were our three different types of genetic markers. RFLP, here we can see RFLP, which is restriction fragment length polymorphism, and this is variation in restriction site where endonucleases can attack. That means here the restriction sites is this and this, so here the fragment will be longer as compared to the restriction site here where the fragment is shorter. Then we have SNP in green. We can see single nucleotide polymorphism, which occurs many times, repeats many times in the DNA, but not tandemly or one after the other. And then in the end we have the VNTR, where we have the microsatellites and the mini satellites. And here these boxes, since they are occurring tandemly one after, that's why it's called as variable number of tandem repeats. Now we have finished the discussion about genetic markers. We are going to see another we are going to discuss another topic, that is genetic markers, sorry, not genetic markers, genetic map. There are two types of map of genetic material or chromosome, that is the genetic map and the physical map. So there are two maps of genetic material, genetic map and physical map. Physical map means we know the exact position of a specific gene on a specific chromosome. In physical map, we know the neighboring genes and it is an exact mapping. For example, if we take a photo of a group of people, then we can know exactly which person is sitting next to which person. So that can be referred to as physical map of genetic material or chromosome. Physical map is the real map of the chromosome or the genetic material, because we know at what locus what is present. Genetic map is a map which is formed by the estimated idea of our linkage study. So in genetic map we know the markers or the genetic markers, and by linkage study we estimate that which marker is closer to the disease causing gene. For example, if a group of people is sitting, and we will estimate that usually Moon sits with Mahfuz and Talal sits with Talha, but exactly we don't have the photo of that group. So then we can say that based on the linkage we are saying and mapping the map of the class. So same like that is the concept of genetic map. Or if I want to draw a painting and I haven't been in the class on that day, and I know that usually Mahfuz is going to sit with Talal and Talha is going to sit with Moon, so I will draw my painting accordingly. So that is going to be genetic map. Whereas in physical map I have the photo, exact photo in which who is sitting next to whom, in front of whom. That is a physical map. Right now or currently, for many diseases, for linkage analysis we use the genetic map. Example, whenever a particular disease is there and we don't know which disease, on which chromosome this disease located, which gene is causing this disease, for that we will estimate the specific markers by identifying the markers and form the respective genetic map. Whereas for some disease such as Marfan syndrome, the physical map is already present, because we know which gene causes the disease, which chromosome number, and on that chromosome where is that gene located. Now we are going to discuss few terms such as coupling, repulsion and haplotype. So now here if we consider these two chromosomes, one is the maternal chromosome, one is paternal chromosome, both of them are homologous chromosome, means both of them are going to be the same chromosome number. So example, these are chromosome number 7 and 7, paternal and maternal respectively. So if star here is the what we call mutant allele and star here is the healthy allele, and M, this round shape is a genetic marker which will be labeled as M, and that is the allele of marker M. So it is first allele and this is the second allele of genetic marker M, so we will label as M2. So if the disease causing gene and M1 marker are what we call closely linked and are co-inherited, they are said to be coupled. So coupling is basically another term for linked. And now what is repulsion? The relation between the star on maternal chromosome and the genetic marker allele 2 on the paternal chromosome, that is the opposite relation. This is called as repulsion. Same goes for this, the star on the paternal chromosome and the genetic marker allele 1 on the maternal chromosome, the relation is called as repulsion. Now what is haplotype? When you have many genes in coupling or many genes linked together on a single chromosome, it is called as haplotype. So this was the concept of coupling, repulsion and haplotype. Now we are going to discuss the concept of linkage phase. So what is linkage phase? In pedigree analysis, if we can determine the relationship between a specific marker and the disease causing gene, then we say that we have determined the linkage phase, or the phase is determined. So now let's look at this. So here in this pedigree we can see that the disease causing gene is present in pink color and the individual is affected, and the genetic marker which we are considering is this blue ball, and it has 4 different alleles: type 1, type 2, type 3 and type 4. So when we get the probability, we see that wherever the gene allele number 1 of the marker is present, the disease is present. So we can say that wherever marker 1 is present, the disease is present by looking at the phenotype and genotype. And so we can say that we have determined the phase or the linkage phase. This means that this, what we call allele 1 of marker is very closely linked to what we call the disease causing gene. Now if we want to calculate the theta, so theta formula was frequency of recombination is equal to number of recombination by total number of offspring. Therefore frequency of recombination, here we had no recombination, it's 0 divided by total number of offspring is 4. Therefore theta is 0, sorry. But now if we add 2 more cases, for example if we add 2 more kids, and since the marker 1 allele the allele 1 of marker was present but the disease was not present, that is very what we call unusual. Whereas the 6th child, he didn't have the allele 1 of the marker but he had the allele 2 of the marker, and still the disease was manifested. So now the theta is going to be like theta is equal to frequency of recombination by total number of offspring. So there were 2 recombination, so that's why 2 divided by total number of offspring is 6, so 1 by 3, that is 0.3. That is the theta value here. So what we understood that up to 4 offsprings the theta was 0, but as the number of offsprings increased, the theta changed. So this means that a small sample may not be accurate. We need larger sample. If we keep gathering information and still after a large sample theta remains small, then we can say that the disease causing gene and the marker are linked together strongly. Now we are going to discuss the concept of logarithm of odds or LOD. So the formula of LOD is probability of linkage to the power of 4 divided by probability of non-linkage to the power of 4 into log, and these results are collected from many families, and the result of this equation is called as LOD score. That brings us to probability of linkage to the power of 4 is 1 minus theta to the power of 4. Why? Because linkage is associated with theta. Because we know that the distance between a disease causing gene and a particular marker is called as theta. So that is why the probability of linkage will be associated with theta. Therefore we write here 1 minus theta, and then divided probability of non-linkage is maximum theta, because maximum theta means no linkage at all, and the chance of no linkage at all is 50% at meiosis. Because if there is 50% chance, there will be 50% chance of co-inheritance and 50% chance of segregation, which is a very high chance. So if we take that case, in that case theta is equal to frequency of recombination, that is 2 kids will be recombinant and 2 will be normal. Out of total offspring 4, that is 1 by 2, so 0.5 is the answer. Therefore our maximum theta is going to be 0.5. So coming back to the equation, so probability of linkage is 1 minus theta, because linkage is associated with the concept of theta, and probability of non-linkage, that is linkage non-linkage will happen, is we have to consider as maximum theta, that is 0.5 into log, and the answer, the result of this will give us the Lord score. Another important thing that we have to remember is that probability of linkage is associated with theta, but the probability of linkage, it varies from marker to marker and gene to gene. This means that, for example, if we consider this chromosome, and on that, if this is the disease causing gene and this is marker 1 and marker 2, the distance between the disease causing gene and the marker 1, or the probability of linkage of disease causing gene and marker 1, theta is equal to 0.01, whereas the probability of linkage or the distance between disease causing gene and marker 2, or theta, is equal to 0.05. So what does this mean? If theta is equal to 0.01, this means that out of 100 chances, there is only one chance of recombination. That means these two are going to get separated. Whereas for M2 marker, theta is 0.05, is slightly bigger. So that means out of 100 chances, there is just 5 chances of recombination. So this explains us when theta is large, the chance of recombination is higher, and therefore the probability of linkage varies from marker to marker and gene to gene, but the maximum probability of non-leakage is always going to be 0.5. So now if we consider the Lord formula and put in the values, so example, if we will consider the marker 2, the Lord score is going to be, probability of linkage will be 1 minus 0.05, because probability of linkage theta, right? So 1 minus 1 minus theta, so 1 minus 0.05 to the power of 4, divided by probability of non-linkage, which is maximum theta, and that is equal to 0.5, like we had calculated, to the power of 4 into log. So that is 0.95 to the power of 4 by 0.5 to the power of 4 into log, and this will give us our Lord score. Now we know what is the Lord score and how to calculate the Lord score and what is the Lord formula. Now let us look at what does different Lord score mean. So if Lord score is 1, 2, 3 or -1, -2, -3, what does that mean? As the Lord score increases, the chances of linkage increases, whereas as the Lord score decreases, the chances of linkage decreases. This means that if Lord score is 1, it means that the probability of linkage is 10 times more than the probability of non-linkage. Whereas if Lord score is 3, it means that log of 1000, so it means the probability of linkage is 1000 times more than that of non-linkage. In short, if Lord score increases, chances of linkage increases, and if Lord score decreases, chances of linkage decrease. Now we have understood the concept of Lord, how to calculate Lord, and what is the meaning of different Lord score. Now one important thing we have to understand is the difference between theta and Lord score. Theta is just the interlocus distance, which is determined by rough estimation by frequency of recombination. So it means that it is just the what we call interlocus distance. For example, here distance between the genetic marker and the disease causing gene. Genetic marker and the disease causing gene, and it is estimated by frequency of recombinations by going through the pedigree charts. The problem of theta concept is that we take into account only one family, and in one family there are very limited number of children, and so the sample is small. The doctors wanted more accurate answer, so they decided that the data should be collected from many families, and this data should be put together to get a more accurate answer, and that can be done with the help of log, and then the data can be added easily. So the Lord score tells us about the likelihood or probability of linkage versus non-linkage of the particular genetic marker and the disease associated gene. So in short, the theta score tells us how close the genetic markers are to each other, whereas the log score actually tells us for the long run whether there is highly likelihood of linkage or highly likelihood of non-linkage. If we talk about a specific disease causing gene and a specific marker associated with it, and we come to know that the log score is +3, it means that this specific disease causing gene and the specific marker which we are considering are strongly linked. Or if there are 999 babies, 999 babies will have this gene present, whereas one will miss out because recombination will take place. So this means that they're strongly linked. Whereas if we talk about another disease which is a specific which has a specific disease causing gene and a specific marker, and the log score comes out to be -3, it means that they are strongly unlinked, that is they have no connection and there is high chances of recombination, and there is no point of considering this disease causing gene with this marker. We have to look for some another marker, because otherwise our analysis will always be wrong. Now we have finished our discussion on log theta, log score, log formula, etc. Now we are going to discuss about centimorgan. What is centimorgan, or the unit of centimorgan? So Dr. Morgan was the first person who gave the concept of interlocus distance, or distance between particular alleles or a genetic marker and a disease causing gene or whatever. So the unit for interlocus distance is named after him and is called as centimorgan. C is small, m is capital. So 1 centimorgan means there is 1% chance that two alleles will segregate separately, and 99% chance that these two alleles will segregate together or co-inherit. Example, if we consider a disease causing gene and a gene marker 1 and gene marker 2, the theta between these two, that is disease causing gene and gene marker 1, theta is 0.01. That means it is equal to 1 centimorgan. If we consider the theta between this disease causing gene and this gene marker here, it comes around 0.05. Theta is 0.05, that is 5 centimorgan. So that is the unit of interlocus distance. Now a question is, how many nucleotide base pairs are there in 1 centimorgan? So in 1 centimorgan there are 1 million base pairs. There are 1 million base pair in 1 centimorgan, and 1 centimorgan indicates that there is 99% of chance of co-inheritance of these two particular alleles and 1% chance of segregation of these particular alleles.